despite the fact that DNA is the same in all cells, that something else must contribute to the fact that the liver is a liver and so on. This is all gene expression. Liver has different genes expressed than kidneys and so on. So, and that means, as a last step, that translation plays there a very, very, very important role. And if you need to remember something from this morning, it is that translation is more important than you think at this very moment. So this 90 minutes or something will, I hope, show you that. So this you know, of course, right? Well, easy. This is the way this entire 90 minutes or whatever the seminar is built up. So it will be very, very, very easy to start, and it will be a little bit more complex every time. So, but I will always come back to this easy and add something more. So that I hope you can, or I don't lose you on the way. So this seems very simple, of course. DNA gets replicated when the cell divides, and when you have some genes to be transcribed, there is RNA made, mRNA transported to the cytosol, and there it's made into a protein. This you learned when you were 14 or something, I guess, 16 maybe, long time ago, and this is what we know, right? Easy. Okay, it is of course a little bit, this is about translation, it is a little bit more complex than just mRNA protein, right? So if you have this pre-messenger ribonucleoprotein, so RNA with all kinds of proteins attached, you get the canonical end modifications of messenger RNA. Like you have your 5' guanosine, which is methylated, and your 250 adenosine, so a poly A tail at the end, and then it's transported to the the cytosol. Right. Just also, this is about translation, about mRNA. Of course, mRNA is just a fraction of the RNAs within the cell. It's it's really minor, the amount of mRNA which is in the cell. The vast majority are different RNAs, non-coding RNAs, right? Ribosomal RNAs, transfer RNAs, microRNAs, and so on. Okay. So, but here we have our microRNA, or messenger RNA. It's in the cytosol, ready to be translated. Well, it's a little bit too fast, because there's different things that can be done. It might be translated, it might just be broken down, or it might be stored for being translated later. And essentially, what makes these, or within the cell, there is a decision made based on essentially a whole lot of other regulatory factors that will determine whether this messenger RNA will be eventually translated. And the most important ones that you will hear about today, when it is about translation, are these EIFs, so the eukaryotic initiation factors. These are the most determining, or the proteins that are most determinant for whether or not an mRNA will be translated. Okay, and once we have them proteins, of course, and that is also nothing new, then of course a protein can interact, can be post-translationally modified with all kinds of stuff. They can be either in the nucleus again, they can be in the cytosol, they can be in the membrane, all kinds of functions. They can be broken down rapidly, they can be stable, so all kinds of these things. But this is very general, the overview. So it's a little bit more complex already than just messenger RNA and in the cytosol protein. So it's more complex than this. So this is taken from a textbook probably when you were at high school. This is also very straightforward, right? So you have your mRNA, you have ribosomes attached to it, the ribosomes scan through the mRNA, and you have a growing polypeptide string, let's say chain, when it goes on and on further towards the 3' end. Easy. And on the way it gets folded, there are chaperone proteins that prevent misfolding and so on. Easy, right? Right. So, for today, what are your learning goals? It is essentially, I will tell you that the beginning, the start, the initiation of translation is absolutely the most important. It's almost everything when it's about translation and regulation of translation occurs at the initiation step. So that's why we will also put most emphasis on how that is done and what then the impact is of this translational control, mostly initiation, on gene expression. All right, and then this afternoon, I think, I'm pretty sure, my colleague will tell you a little bit more on how we use that in research and what we can do and what we have done in this area. So, I told you, it's really easy. 3 steps. You have your mRNA, of course, you have to start, you have to process, and you have to end. Okay? Good. That's what is shown here. Here it's already a little bit more some added complexity, because your mRNA is not just, it just doesn't start at the beginning. No, your mRNA has a starting codon, has its coding region, but there's also a large untranslated region both at the 5' and at the 3' end. So in these UTRs, which are here in black, you can already sort of imagine they're not there for nothing. All or most of the regulation takes place exactly in these 5' and these 3' untranslated regions. Okay? So we will see that this will be very important. And also we know the start codon, which is methionine. And of course you have methionine at the start, but there are also methionines within proteins. So in the vast majority of the cases, not always, your translation will start at the first methionine it encounters. Okay? So comes back later on. Okay, so the first, whatever, or the next 30 slides will all be on translation initiation. So, how does it start? You have your, essentially, here your 5' cap, and that is a very important signal for cap-dependent translation. And the vast majority of translation will be cap-dependent. So it starts here. Here, where is my mouse? There. So it will start here at the cap. And it will start with the 40S ribosomal subunit that will find this cap. I will tell you how in a minute. That will scan, scan, scan until it reaches the first start codon, so your methionine. Then the 60S subunit will join, and together these form the 80S subunit, and they will then start scanning and do the real job. Anyone an idea what this S stands for, by the way? And what is that Svedberg unit? Why is that 60S or 40S? What do these things mean? How is it determined? Any idea?
Sedimentation coefficient.
Right, good. So it's a sedimentation coefficient. So basically you run these things in a sucrose gradient, and the deeper it gets, so in the sedimentation gradient, the higher the number. So the 40S runs less deep than the 60S. And if you combine these two together, they run less deep than the sum of the two. So that's why 40 and 60 is 80 and not 100. All right, so that is what is happening here. So here, if you recruit then here the full ribosome, then there is elongation, and at the end, I assume, so elongation, also this. I will absolutely say nothing but this about elongation. This is again from textbook. So what happens is your transfer RNA comes in loaded with your amino acid, with an anticodon, which is complementary to your codon, and then it's added and so on. And then it falls off, the next one comes in, and your chain grows essentially. So that is what is going on. Very important though for elongation, or in general for translation. And you might remember that from last year when I talked about hypoxia, I think, is that 75% of all energy in a cell is used for translation. So maybe you forgot that from last year, but then I say it here again. So it's the most energy costly process in the cell for sure. So if a cell encounters stress, like hypoxia for instance, which very often occurs in solid tumors, it will react by immediately shutting down overall translation to reduce its energy, because it will not have the energy very often. in times of stress, in times of nutrient stress, for instance, also. Then the cell will start blocking overall protein synthesis for this reason, because it's just way too costly in terms of energy. And then at the end, also very straightforward, then you have a stop codon. There is no transfer RNA with a stop codon where it's complementary, and instead there will be a release factor that will be put in place, and then the entire machinery falls apart and it's over, done. All right, so termination, elongation, once it's started, that will normally all go well. So there's not much to be regulated there. It is all about initiation. This is the rate limiting steps. And as I said, so the reason or the way it's regulated, it's through all these eukaryotic initiation factors. And that is pretty complex. And I'll show you that it is, but in a, I hope, pretty comprehensive way. So why is it? So this is already a little bit more complex. So you have your 5' 3' end, you have the start codon, and we know already that the subunits are being formed at the cap structure normally. They scan, and then they start making the protein. But in this 5' UTR and in the 3' UTR, you see all these kinds of modifications. There are, of course, the canonical ones, the cap structure and the poly A tail, but also these kind of things. Irises, there are hairpins, there are sometimes binding places for proteins within this 5' UTR. There can be upstream open reading frames, which is an open reading frame which is not the reading frame that the protein is supposed to use, and so on. And this can be also in the, to a lesser extent, but also in the 3' UTR. Of all these things, I will give examples on how the cell will use these, or how these things are being used for regulating translation initiation. All right, and also pretty interesting to know is that these mechanisms will all prevent translation initiation from happening. So that means that the standard condition in a cell is translation initiation, so translation is on, unless there are some mechanisms within the 5 or 3' UTR that prevent it from happening. But normally it's on, it's ongoing, right? That's of course a little bit a very strict conclusion, because of course it's not just these structures that will help or will determine whether or not RNA will be translated. Much more, it is also these translation initiation or these eukaryotic initiation factors that will determine whether or not it will take place. Because if you don't have enough, then the structures might be there or whatever. You need first of all these factors to start. All right? So that is very important. What are these translation initiation factors? Here we go. Right, so you have here the cap structure. So this is the 5' end of your mRNA with the cap structure. The first one, and a very important eukaryotic initiation factor that binds, is EI4E. So EI4E is very highly regulating, and mostly when the amount of EI4E will determine whether or not translation will continue. So this is mostly the rate limiting factor, it's EI4E. If there is not enough EI4E, the rest will not, cap-dependent translation will not take place, okay? Of the others, there's mostly, they are more abundant. So they are mostly in high enough amounts present for translation to occur. So the EI4G, that's the second one. This is also, of course, a very important one. And why? Because this serves as the docking station for putting all the others together. So this has very, so I will show you in a minute, binding sites for all the other eukaryotic initiation factors for other proteins that will make sure that the complex can be formed. It holds it together. It serves really as the docking station. Then we have our EI4A, which is a helicase, which will make sure that the secondary structures in the RNA will be unwinded when it scans through, so that it gets not stuck when there is like a hairpin and so on. So that is an ETPase. And then you have PABP, which is very neatly, or its name, it will come back. These people that discovered all these proteins, they give it these really fancy names. PABP is the poly A binding protein, which makes sense to a certain extent, I guess. So this is very important. So this complex is absolutely crucial for cap-dependent translation initiation to occur. So this, I think I just said, this is not so, this is just some additional information. Just EF4E, very important. EF4A is an ETPase, so we'll use this energy to make sure that it can run through this 5' UTR. And then here, this is very important, this is EF4G. You see that this is the docking station, and it has, this is a schematic drawing of how it looks. So it has a binding site here for EF4E, for EF4A, so the helicase, and for BABP. And it has also, that will come back later, binding sites for some kinases. And overall, right, so when on EF4G, A and E and PABP are bound, then we call this complex EF4F. For some reason that I don't understand why E plus G plus A is F, what the logic is, I don't know, but that is the way it is. So if you talk about all these eukaryotic initiation factors, you have to be a little bit concentrated that you use the correct names. Anyway, and this is the poly-ABP, and you see immediately why this is so important, because it will give some structure. So it will sort of keep the start and the end together, so that it's not just floating around there. If that happens, then translation will not occur. So this is very important, so it has also some structural role. All right, so this is the first step. EF4F has to be formed at the cap. Right, then the next step, of course, is you have to, because here there's no ribosome at all involved. So you need to form now, that's the next step, your initiation complex. So your first amino acid has to be brought in somehow. And there's another factor, which is EF2 in this case. So there are two very important factors, that is EF4 and EF2. And then with the letters. So EF2, very important because this carries in the first methionine. If this doesn't work, nothing will happen, right? So you see, you feel immediately already, intuitively, that if there is a regulation on protein translation, that the factors most likely to be of importance are EF4E and EF2. You already can sort of intuitively feel that. So how does this? This is a schematic. So this is EF2, and this carries in this ternary complex the methionine, and it is a GTPase, so it needs GTP, and the GTP needs to be converted, or there has to be a release of energy once the complex is formed. That will come in just a sec. So this is the role of EF2. It will load your first methionine onto the 40S ribosomal subunit. And then together with some others, so EF3, EF1, and so on, they all help scanning later on. So these are all other eukaryotic initiation factors that will together make sure that this will happen, so that this 40S subunit will find the start here at the cap. And in this case, EF3 is very important because EF3 will dock to EF4G. So you see immediately that this complex is formed, and that now, once this happens, and this is a process that requires energy, that once this has happened, then it can start. Only then. So all this complete complex has to be formed. But once that is done, we can start and scan. And what happens then is that this 43S complex, which is the 40S subunit with all this, with the ternary complex, with EF2 and these other initiation factors, will then start to scan for the first methionine codon, right? And all these other ones, they are essentially helping them to do so. And then once the starting codon is found, then the 60S subunit comes in, they join together, and they form the ADS complex. Essentially what happens first is when the 40S or the 43S complex reaches the start codon, then a stable complex is formed, and then it is from a 43, it is the 48S complex. And then this 48S complex, then the second subunit comes in, and your complete initiation complex, which is the ADS complex, is formed. And then when that happens, all the initiation factors that were needed to go from here to there, they will then fall off. And essentially this also requires energy which is bound to other initiation factors, so you can already sort of start to feel how much energy you need for all of this. Because for every single steps here, ATP or TTP is needed. All right, so this is essentially a summary so far. So we started off with this sort of picture, where we said, okay, there's a 5' end and the ribosomes are put together, then you make this protein, and then there's a 3' end. I think that here, this one shows a little bit more complexity, and that it's much more like this. Okay? Clear so far? Good. You're so quiet. So, and that is really neat. You can see that here there are many proteins involved. So it's a pretty complex process. So you can already feel that there's many ways that you can use here to interfere with this process. And if there's many ways to interfere with that process, the reason why a cell does that is because it will allow it to very accurately react to signals from its environment, right? So to very fastly fine-tune a response. And that can be very much different in a liver than it can be in your brain or in a tumor versus in a normal tissue, for instance. All right? And this is, of course, you already see that it's involved in everything, from the start till the end, from embryonic development until we die from cancer, right? And essentially, next to translation, and I hope for your colleagues next year, like all these things, how it's involved in translation initiation and all this differentiation, there's many other pathways and so on, like for instance the Notch or Hedgehog pathway, which now we don't hear too much about, but which we investigate, for instance, now also in the lab. There's a group with like 7-8 people doing nothing but Notch research, which is also very important in the entire embryonic development, differentiation, metabolism, cancer. So it is really very interesting field, because it tells you, or it sheds light on how it's possible that we start from one cell, essentially, when we are being made, and that yet we sit here and we look all different, and yet in every single cell you have the same DNA, and yet it is so different, your liver, your brain, your skin, whatever. And that is, translation has a major role, but there's much more important pathways, like this Notch, Sonic Hedgehog, and so on, that are very important. And I hope from next year, for instance, that you can also include this in your, well, then maybe not for you anymore, but definitely for the ones that come after you. Anyway, that being said, that was a little appreciation of my frustration that you do not do it so far, but it will come. Anyway, so there is then, when we have translation initiation, so believe me, elongation and termination is not regulated at all to this extent. So we have translation initiation, control is taking place at this level. There's two ways, or two different kinds of regulation. Either the control is global, so it is overall translation initiation that will be affected, or it can be very specific. For one mRNA will be differentially translated than another one. And that is regulated in a very specific way. All right? Of all these, I will give examples. And it's nothing but examples. The main point for you is to understand the difference between global and specific. I will never, ever ask that you have to explain me a very specific example that I will give you. So forget it. Just pay attention and that you feel the importance or the difference between global and specific. All right. So yeah, this is what I will tell you. So it's, as I already showed you, it's mostly global regulation of initiation will occur through the initiation complex. So EF4F, EF4F being E, G, and E and A, right? Or via EF2. And I will show you of both of them some examples. Here, so global translation initiation regulation through EF4F. One pretty obvious way of doing that in a cell is, of course, EFE first has to bind to the cap structure. If that happens, then EF4G comes in and so on, and the complex can be built up. One obvious way for a cell to regulate this is, of course, to prevent 4E from binding, right? So to take it away, to compete with EF4G for binding of EF4E. So how does a cell do it? Well, a cell has a protein which is, again, very elegantly, logically, and very excitingly called 4EBP, which is the 4E binding protein, right? So so translation regulation is very often regulated through the availability of this protein EF4E. And what happens? So 4E can be bound by 4EBP either when it's already bound to the cap or when it's still free in the cytoplasm. And when it does, when 4E is bound to 4EBP, 4G can no longer bind. All right? So this is a competitor for 4G. And if it cannot bind to 4G, translation initiation cannot occur. So and how is this process regulated? Well, that is regulated through phosphorylation of 4EBP. If 4EBP is phosphorylated, then it can no longer bind to 4E, right? And if it's hypophosphorylated, so if it's not phosphorylated, then it can bind. And the cell will regulate, depending on the conditions that it's in, that either 4EBP will be highly phosphorylated or not. So, can you, without looking at your slides, which you might have printed, come up what kind of a condition? When would, for instance, 4EBP be highly phosphorylated? In what kind of conditions? In? Right. Yeah. In energy-rich conditions when everything is in place. For instance, also, what do you think in cancer? Would in cancer 4EBP be more phosphorylated or? Well, it's a tricky question actually, but it's a good one. In cancer, we have a solid tumor. Would 4EBP there be mostly phosphorylated or not? Every answer is right, by the way, so it might be yes and no. So it might also depend on the conditions in the tumor. Let's say under hypoxic conditions in the tumor. Oh, I gave this away. Under hypoxia, so we have a tumor, which we know, of course, you are very, very much aware of the fact that these are hypoxic very often. So in these hypoxic conditions, would 4EBP be hypo or hyperphosphorylated? Who says hypo in hypoxic conditions? So hypo means, so there is not much phosphorylation, so it will bind to 4E. Yeah, I'd say again, yeah, okay. I saw a few hands, which I would say, yeah, I would also say hypo. Because under hypoxia, I said there is not much energy, so the tumor will shut down its overall translation. And hypophosphorylation of 4EBP, which takes away 4E, will prevent translation from happening. So this is one mechanism that a cell can use to downregulate translation, right? So instead of learning all these examples that I will give you, this is more the kind of things how you should look at this, okay? So this is hypo. When would it be hypo? When would it be hyper? Does it make sense? Why does it make sense and so on? All right, so if I would ask you, in a tumor, like I could put a sort of a saying, let's say, 4-ABP is hyperphosphorylated in solid tumors. Agree or not? Why? Why not? These kinds of shit questions I can ask you. But you know, I'm not saying that it will be like this, but it's more like, at least in that sense. All right, so here it's then explained, so it will be increased for EBP when there is like signals from the cell coming in, that it says, okay, grow, go on, you know. And in hypophosphorylation will take place when there is not much energy, in hypoxic conditions, for instance, when there is not much nutrient, and in all kinds of stresses, when one of the signals of the cell will be to take low profile. That's basically it. For EBP, and this is interesting for your journal club or the paper that you're reading now, the way that for EBP phosphorylation is then regulated. By the way, you can always, very easy, that's the way I do it at least, remember that hypophosphorylation blocks translation. So that all comes back. This is the, so we can never, hyper is translation. It's the word. Stupid, but just that you do not miss it. So 4-ABP phosphorylation in itself is then of course also highly regulated, and in this case, this is through mTOR, and this comes all back in your journal club or your assignment. So I will not go too much into detail because then everything is already solved. But what happens essentially is mTOR here, mammalian target of rapamycin, stimulates, when mTOR is active, it stimulates translation. So it will phosphorylate 4-ABP. Phosphorylate 4-ABP does not bind ER4E, so translation will go on and continue. mTOR also phosphorylates S6K. S6K is a very important kinase, which will then, or a protein kinase, that will make sure that is involved in processes of making these proteins which are important for translation. So all these ribosomal proteins and so on, it's made through S6K and all these elongation factors. So if that is highly active, you have all the tools, everything is in place in sufficient amounts to cope with this increased demand for translation. That's basically it. All right, and how is it regulated? So it might, so this is essentially, there is two major ways. We have either receptors that are on the cell surface, and there's a signal telling them to grow, or in tumors, very often it is deregulated here, so you have deregulated signaling, or PI3 kinase is mutated or whatever. So when this is active, AKT will be activated, and when AKT will be activated, and this is funny in a way, it will phosphorylate TSC2, and normally you probably think like, oh, when something is phosphorylated, it's more active. That's very often, or well, very often, that is in very defined cases, not the case. For instance, here, if AKT phosphorylates TSC2, it's an inhibitory phosphorylation. And TSC2 discomplex will normally inhibit mTOR. So if you inhibit the inhibitory complex, the inhibition is gone, right? So that is a little bit, so if you, it's hard to follow, then you just sit back, relax, and look at it, and you will understand. So you take away the inhibitor, so it will be active. And then the other major pathway that is involved is AMPK, which is a kinase which responds to changing levels of energy, which is an energy sensor in the cell. And if ATP goes down, AMPK will go up, and AMPK will also phosphorylate TSC2 complex. But in this case, it's not an inhibitory, but a positive phosphorylation. So it will increase the strength of the inhibition here of mTOR. So then it will stop mTOR from being active. In other words, if ATP is low, under hypoxic conditions, for instance, or in times of nutrient stress, ATP will be low, AMPK will be up, and AMPK, through a stimulatory phosphorylation of TSC complex, will block mTOR activity, which is exactly the opposite of AKT. So in other words, also when ATP is high, AMP key will not be activated, will be downregulated, and this inhibitory phosphorylation will not take place, right? Good. So, also maybe important for your German club or your assignment is, of course, if you have 4-EBP, but in a tumor, one way to get around it is to make lots and lots and lots of EI4E. And if there is way excess of 4E compared to 4-EBP, then all the 4-EBP that is around might bind 4E, but there will never be enough 4-EBP to bind all the 4E. So just by making way, way, way more 4E than there is 4-EBP available to be bound is a way for a tumor to get around, because there will still be enough 4E to initiate translation. Right, and that's more or less what is in here. Okay, then another way of regulating globally the EI4F initiation complex is through phosphorylation, and this is actually something that is, as the models are currently understood, that is something that predominantly is of importance in tumors, not so much in normal tissues, but especially in tumors. And that can be through stress conditions or through growth factors and deregulated signaling cascades. And then these kinases, which can also dock onto this EI4G, can phosphorylate EI4E. And when EI4E is phosphorylated, the idea is that it's more, that it will increase translation more, that it's more efficient in initiating translation. And one of the reasons, which I don't mention here, and which is probably also not so super important, but there is also, well, it is important, but well, it's not that you need especially to remember, but there's another protein, which is the 4ET protein. And instead of the 4 binding protein, what should the T stand for? Guess. 4ET, T. What would be, how can, this is about, it's the same issue, it's about availability of EI4E. So EI4E can be bound by another protein, preventing it from being bound to the cap structure or to EI4G. This will also, this 4ET limits availability of 4E. So what could another way of binding a protein away? What could be another way of getting rid of a protein? Yeah, it's straightforward. You don't have to think too much. Yes?
Not translation of 4E?
Yeah, no, it's not, no, that's not it.
Transport?
Yeah, well, transport, transfer. Right, so taking away from the site, from the place where it's supposed to do its job. What 4ET does is bind to 4E and transfer it to the nucleus. But in the nucleus, it cannot take part in translation, of course. So, and when it's phosphorylated, this transfer, this binding of 4ET and 4E is prevented. So that's probably one of the reasons why it can stay then much more in the cytoplasm and when it can then take place in translation. All right? Good. If something is not clear, you have to yell or to raise your hand. Okay, and then another way, so there is essentially, you take it away, you prevent it from being there by binding or by transporting it away. The other way, it's also obvious, is to degrade it, to make sure it's there, and just instead of being stable and being able to be used, it's degraded by proteolysis. And that's also what is happening very often, not for so much for 4E, but especially for 4G and the poly-A-B, the PAB protein. So this can also be degraded, and when it's degraded, yeah, it cannot be used, of course. Sometimes, and that's neat, of course, that happens in a cell when it's infected by a virus. A virus will make, or the proteases that come with the virus, can very often specifically break down these proteins. And the reason for that is that then your cap-dependent translation in your cell will be down, and then the energy that remains, the virus will use for its own replication through another mechanism which mostly involves, we think, irises, which I'll come back to in a second. which is a very intelligent system. A virus is nothing but a strain of nucleic acid, but yet the way it infects a cell and uses it to its advantage is very, very intelligent design of Mother Nature. Okay, so this is a little bit an overview, and as I said, okay, for E, it's very often deregulated in cancer, and when it's deregulated in cancer, of course, you can also think of ways to target it. All right, I don't know, is this slide in your thing or not? Is that in yours? Yeah, okay. Well, I put it recently there. Anyway, so this is, you can look here, and I think it makes sense to do so. Okay, so we had EI4E or the EI4F complex, that's one, the cap-dependent complex that is formed there. Then the other one is EIF2. EIF2, we said it's also very important because this one has to bring in the ternary complex and your 4DS subunit to this EI4F complex, right? So you can also imagine that messing around there can help to regulate translation initiation. So a little bit more into detail, I told you that it would be more and more adding complexity. This is again the same complex where here you have EF2 with the GTP bound and with your methionine transfer RNA. When there is translation initiation, I told you, okay, when that happens, the GTP that is bound here is converted, hydrolyzed into GDP. That's an energy costly process. And then, of course, that has to be recycled for the process to be able to continue. And this is something that is done by EF2B. So EF2B is absolutely crucial for recycling of the GDP for the GTP, for the exchange. And when that is happening, EF2 can again be loaded with methionine and can load more ribosomes, let's say, onto the mRNA, and translation will be more efficient, right? Very important. How does a cell now, or what can happen, is that there are kinases, the so-called EF2 alpha kinases, the ones that phosphorylate EF2 alpha, right? That can phosphorylate EF2. And when EF2 itself is phosphorylated by one of these kinases, EF2B can no longer function, can no longer do its job exchanging the GDP for GTP. So it prevents the ternary complex from being loaded with GTP. And if there's no GTP, it will never ever be able to be part of the initiation complex and start scanning, right? So this is a way for a cell to inactivate EF2. And this is this is done here at EF2 alpha, right? I told you it's more complex. So EF2 has 3 subunits, alpha, beta, gamma, and it's EF2 alpha that is phosphorylated. And if you then, on the serine 51, and this is, when this happens, so you have an inactivation of this. And this is a tool that I think Casper will talk to you about this afternoon, that we can use in the lab, because we have EF2 alpha, in which you can modify the serine 51, and you can convert it into the serine into an alanine. And an alanine cannot be phosphorylated, or it mimics, it mimics phosphorylation. So in other words, it's always inactive. Or you can also do it the other way around. I have to think whether what I say is right or wrong. But anyway, so you can mimic, or you can play around with this, and then you can mess up your translation efficiency, essentially. That is what it's all about. That's the main message. So, and what are these kinases? These are things like here, HRE, GCN2, and so on. Don't be bothered too much about the names, but you can already, and I'll come back to that in a second. So overall, when these kinases are active, since you tackle EIF2, you see that EIF2 loading the first amino acid. Yeah, that's something that has to occur for every single protein, of course, to be made, so that is a global way of inhibiting translation. All right? Maybe this is an interesting one, PERK. Does anyone remember anything about PERK from last year? I know you should, because I teached. Many often people that are not native English say teached. which is not right. It's thought, my Canadian colleague always told me. So I taught you last year something about PERC. What did I teach? Who remembers? It must have something to do with hypoxia. If you would have yelled like, oh, it has involved in hypoxia, I would be so super happy. So maybe, so anyone an idea about PERC, in which process it is involved? Did I hear hypoxia? Yeah, right, good. I'm so glad that you remember. Just I'm sure that you don't remember the details, so I will briefly mention it. Because under hypoxia, we all know HIF and the transcriptional regulation, of course, that you remember. But the translation, I told you last year, like next year, I will give you an entire seminar on the importance of translation. And look here under hypoxia what happens. And that it's where PERC came in. So what happens under hypoxia is that translation is in general shut down, and a very important part comes from the activation of this PERC kinase. Because under hypoxia, when there is no energy, the proteins that are being formed will be not properly folded. And this, in the endoplasmatic reticulum, will cause unfolded protein response. It's always so straightforward. So if proteins are not properly folded, this will give a response. And this UPR will trigger PERC. So we'll activate PERC. And what PERC does, now you know, last year you didn't know, but now you see the ins and outs. PERC will, amongst other things, phosphorylate EIF2, and by doing so, shutting down translation, preventing more proteins from being made, and thus more proteins from being improperly folded. So that is the way the cell deals with all this shit or whatever. Things. Okay, good. So globally, EIF2, EIF4, primarily or almost entirely at the level of initiation. But we know that not all, well, that's also for globally, but especially this is the bridge to the specific regulation. So not all mRNAs will be affected equally. I'll come back to that in a minute. And this I more or less told you already on the way. The reason why it's so important to control, or why it's so efficient to control, is because this will go very, very fast. And it will allow you, all these different ways of regulation will allow you to have good and quick response and fine control. That's essentially. Right. Now, the next part will be basically showing a few examples that instead of globally downregulated translation, some mRNAs might be, in very, very specific cases, be shut down, will be prevented from being translated, while all the others around it will be happily translated. All right? And there, all these different 5' and 3' UTR modifications come in. I will give, I think, an example of all of these which are listed here. So, and that is, again, there's three ways. The last one is probably related to the A and B. Two ways, either what mostly happens is it prevents 43S recruitment on this specific mRNA, or it might be still recruited, but it will be prevented from functioning properly. These are the two ways. So, of course, one of the things that you can imagine of such a mechanism is just to sterically prevent it or hinder this complex. And that is shown here. Again, these are examples, right? So don't ever waste time on memorizing all this and what IRP and so on just involves. This is more overall the kind of methods, the kind of mechanisms the cell uses. So in this case, it's an example of this protein, which is called iron regulatory protein, which can block here the translation of this protein called ferritin. So the mRNA here is the ferritin mRNA, and what is ferritin? Ferritin is a protein which is involved in iron storage. Alright, so when ferritin is there, it makes sure that the iron that is available is getting properly stored. Now you can imagine in conditions where the cell needs iron, ferritin is not supposed to work because instead of storing it, the cell has to use the iron. In that case, the IRP, the iron regulatory protein, comes in and it will bind to one of these secondary structure in the 5' UTR of ferritin, bind there, and when it sits there, whatever the cell will try to do, the 43S or the complex here, and the 43S, where normally the 43S complex should be, with the EIF2 and so on, that simply cannot go there because IRP is in its way. So it simply sterically hinders the formation of the complex. And it makes sense, of course, because then ferritin cannot be used, and the iron that is needed in cases of stress, when there is not enough iron, for instance, so at least the iron that is there will not be stored but can be used. So that is the idea, steric hindrance in this case. Another one, and this is just an example showing that it might also be dependent on structures in your 5' UTR. So this is just not more, not less. Sometimes they are, it depends where they are, or it depends how big they are and so on, and that they can then hinder 43S recruitment. So that is essentially the same. So in this case, it's more the steric hindrance caused by this secondary structure. And this might then be and dependent on whether or not your methionine codon is there, and this might prevent the 4A helicase activity from unwinding this. So that is another way of doing it. And then, also very interesting, because these things, just very specifically and without going into details, this essentially is a mRNA specific 4EBP. That is what this meskin is. So it will specifically bind to 4E, preventing it, or thereby preventing 4G from binding to it. So it's a competition for 4G again, but instead of globally, it will be only happening in this specific case. In here, it is for this specific, whatever, I don't even know. Oh yeah, for this, this oocyte maturation. So that happens in some cases when you don't want, or you just do want oocyte maturation and so on. That doesn't matter at all. So but what it is you have a regulatory element, CPE, to which CPE protein can bind, right? And this can also bind to meskin. And when these two are bound, 4E is being bound by meskin, and 4G cannot bind. So meskin is a mRNA-specific 4EBP type of thing, right? So this also happens. And then there is another one, which is essentially kept independent, and it's also important to know that many of these processes, we don't know the exact details. So you will see, the further we get, that there's many more question marks coming, because we don't know, but this is what we see. So you have here, in this case, in Drosophila, this is happening, and this is something which has to do with maternal, paternal dosage inhibition and so on. But we don't know exactly, but what happens is that you have this poly-U tract, both in the 5' and the 3' UTR, and this other protein is binding to it, and this prevents the scanning of the 43S. We know that it's happening, but exactly which factors are involved, we don't know. So probably here there's some other factors that will prevent this from happening, but at least it's again a sort of mechanism. So there it's not located around the cap, because these proteins, they bind far away from the cap in the 3' and the 5' UTR, yet they prevent the 43S from doing its proper job. Right, and then some other things, that's the second part. You have a situation in which the entire complex can be formed, right? 43S, it's loaded and so on, and then it scans up to the start codon, But then the step for the 6S complex or the 6S subunit to come in is prevented. And this is in this case a very specific protein, which is the LOX protein. And here you have these, again, these specific sites in the 3 or the 5' UTR, in this case in the 3' UTR, that will prevent this from happening. But you see, it's still, here again, there is no question mark, but also here we don't know exactly how it works. So there's probably other initiation factors here that play a role. So, but the overall, again, this is more like in your head that you try to think of ways that this might occur. Another one, which is important, but of which we know much less than we do know, is the use of upstream open reading frames. So what happens sometimes in a protein, you have your open reading frame, which is the one that is supposed to be the full protein. But to prevent efficient translation of this protein, in some mRNAs you have these upstream open reading frames, which are non-functional, that they will prevent essentially the actual open reading frame from being efficiently translated. So, and how does it do that? I'll show you in a minute. And this is an interesting example because, for instance, when you have stress, nutrient stress, there is no enough amino acids or there's not enough building blocks to make your amino acids and so on. Of course, when you don't have the building blocks for your protein, you cannot make too much protein. So a cell will then overall, globally, downregulate translation because the bricks are not there to make your protein. So this is happening very often through this GCN2. GCN2 is one of these eIF2 alpha kinases. So eIF2 alpha is phosphorylated, global translation is down. Yet, what does the cell do? It will now increase specifically these mRNAs, such as GCN4, which is a transcription factor that is needed to make more amino acids. Again, a very neat way of Mother Nature to fine-tune this entire system. So there is not enough amino acids, global translation initiation or global translation breakdown. But these genes, these proteins that are important for making amino acids, which there are not enough, these one will be specifically upregulated. And in this case, this is an example of upstream open reading frames. And how does it work? I'll try to show you essentially. So here in this GCN4 5' UTR, there are 4 upstream open reading frames. And so in cases when there is lots of amino acids, so everything is peacefully and these cells are very happy and growing, you have your complex being formed, right? And you have your initiation complex, and it enters the first upstream open reading frame. There your ATS complex is being formed and it will scan. But it will scan, or very soon, it will turn or run into the stop codon and it will fall apart again, right? So this, because normally that is what a cell does. When it encounters its first methionine, there it will start scanning, and there it will start translating. So but here, when it enters, when the first upstream operating frame is done, it will fall apart. And so it has to start again. Only a fraction of the 40S subunits will continue scanning along. All the other ones are lost. So you see already, efficiency-wise, you lose a lot here. So and then the chance, since there is lots of methionine available, right, the chance that this happens again, that it's loaded into a full complex again before it runs into the proper open reading frame is pretty high, because the components are there, right? So the chance that it runs into, and apparently it does so if it runs again into, or if this complex is formed again, mostly in this upstream of this open reading frame 4, right? And then when it encounters then again the stop codon in this upstream open reading frame, it will fall apart again. And when then that happens, mostly in the vast majority of the cases, there will be no scan again or formation of this proper complex again at the proper operating frame, right? And the reason for that is that you have that much excess in ternary complex, that the chance that you will have a properly loaded complex before the last operating frame is pretty high. Okay? That is important. I think I have all blah, 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 so I said that. In case you have eif2 alpha phosphorylation, most of the ternary complex will not be formed. There will always still be a little bit, there is always some basal eif2 alpha will be available. So, but relatively speaking, the chance that you will have more than one reloading and completion of this complex before you run into the proper operating frame is, relatively speaking, much less, right? So, in other words, the 40S can scan and has a higher chance to get past these 4 operating frames before it will enter the right one. So, relatively speaking, there is a high, so this is shown here in the arrows. So the chance that this is happening is much higher than this, and here it's the other way around. And that's the way the cell does this. You have to think, if it's not so super clear now, then sit back and look at it, and you will see what is going on. So it is a matter of the chance of this being formed in this area is much higher than it of being formed in this area, just because you have here much less to be loaded onto this complex. Anyway, it's just another example. And then here, irises. What are irises? Irises are internal ribosome entry sites. In other words, the ribosome, the site of entry of the ribosome is not in this case the cap, but it is this this iris, right? So the complex, your initiation complex, will not be formed at the 5' cap, as we've seen here. So it will not be cap dependent, but it will be cap independent translation, right? So that is a very specific secondary structure of which we essentially know nothing. The only thing is that we know that it happens, that it is there, that there's lots of genes that we know that have it in their 5' UTR and that can use it, but there is no kind of consensus how a iris should look. If you look into all these different genes, there's not something that is very much common. So we don't actually know what is really happening there. The only thing that we do know, and which is for this context very important, is that you don't need your EI4E. So even if EI4E is not there, you can use it. These are essentially things that viruses very often use as for their translation. All right, so instead of this is the normally cap dependent way of translation initiation, so just at a cap. So here a very similar structure is being formed, not with EI4E, but with some other factors probably that we don't know. But this is what we know is happening. So there is no common structure in these proteins. The only thing is that we know, which is common in some mRNAs, is that they can have cap independent translation. That's essentially it. So these are ways that you can do that in the lab. So if you would make a construct, just don't look at your thing, because I think the solution is on the next slide. So if here you have a cap dependent translation, so an mRNA or an RNA with a cap, with here an operating frame, here is an iris, and here's another operating frame, and here's your poly A, right? This codes for luciferase, and this codes for beta-galactosidase. And you can stain, or you can then, in the lab, stain for both, or check the activity of both luciferase and beta-gal by adding the correct substrate, and then you can do the proper readout. So what experiment would you do if there would be a non-functional iris? So a non-functional iris, what would you see in your experiment? Would you see luciferase, or would you see beta-gal, or would you see luciferase and beta-gal, or what do you think? Who says, if I have a non-functional iris, so this is the iris, and I tell you, okay, we put here like some kind of structure which resembles an iris but is not an iris, and we do the test, and we want to see what is here being translated, right? And I ask you, okay, would I see then in these cells luciferase activity, would I see beta-gal activity, or would I see both? Who says I would see both luciferase and beta-gal activity? Okay, some, okay. Who says I would see only luciferase activity? Also some more, I must say. And who says no, we will see only beta-gal activity? Okay, also some. I'll think I would see only luciferase activity. The reason for it is, if, where is my mouse? Here you start cap-dependent translation, and here, so you enter or you meet a stop codon, and when there is a stop codon, everything will fall apart. So, and the only way when beta-gal should be activated or should be expressed and translated is when there is also an initiation complex formed here, cap independent wise. And this can only be the case if you have here a functional iris and not a non-functional iris. If this is a non-functional iris, this will just be of no importance at all. All right? Good. Translate in vitro, you get this. What would you see? Oh, if not, I just asked that, so you've written that down. Okay, and these are just a few examples of genes that we know that have an iris, but if you would look, and what they are important for, but if you would look into their consensus sequence, you would see nothing at all. So in here, I will essentially not talk about microRNAs at all, because this would be a 2-hour lecture on its own. The only thing is that we know that also other RNAs, microRNAs, other microRNAs, can prevent translation from happening. So microRNAs, they can either be, can lead to yeah, it's way too much. So just remember that microRNAs can bind two regions in the 3' UTR, and can then either prevent the formation of the complex, or can be, see here also again a question mark, somehow interfere with the scanning of the complex before it enters or encounters the stop codon. But it is yeah. microRNA can also lead to mRNA degradation and so on. Can also lead to perfect mismatch, which is then RNA interference. But this is too much, so it will blur your view. Just remember that microRNAs can somehow also block translation initiation by interfering with the scanning of the complex. Okay, so I think that I showed you that mRNAs are not created equal, that some are being more or have the potential to be more regulated than others, that they're also not seen the same, that's a consequence, by the ribosome, and so that when you have limiting factors, that the ones that are needed most, the strongest one, will win, which is very, very neat for a cell to survive in terms or in cases when it will be important to survive. So that is all building material by Mother Nature and evolution. So how, and I think Casper will talk much more about this afternoon, how do we investigate this translation? Well, this is the way to do it. You have a mRNA here, and an mRNA, if you want to look at how efficiently a certain protein is being made, or how efficiently a certain mRNA is being translated. Well, you can do this by making a distinction between these kind of things. Because an mRNA which is not efficiently translated, there will be just one ribosome attached to it, or two. So that will scan. And then you have your peptide or your protein being made. If a protein is being very efficiently made, or an mRNA is being very efficiently translated, it will be continuously loaded with new initiation complexes, and there will be, over the entire length, many 2, 3, 5, 6, 7, 8, 9 ribosomes at the same time scanning the same mRNA. So, and of course, if you have more of these ribosomes attached to it, and you run it through a sedimentation gradient or sucrose gradient, and you spin it, then the ones that are heavier, those are the ones with more ribosome attached to it, will run deeper, right? And these things, when you do that, so you have your RNA, you load it on top of such a sucrose gradient, you spin it in an ultracentrifuge, and then you can scan for your RNA, which comes out here at the bottom. So you just scan it, and the machine will give you a readout, and that will look like this. This is a typical profile, what you get when you do a sucrose gradient and looking for polysomes. So what you see essentially here is your 40S, your 60S, and your 43 or your ADS complex, which is just at the start. And then here you see mRNAs with 2, 3, 4, 5, 6, 7, and so on ribosomes attached to it. And you really see these nicely curves. And then you can isolate, if you want, here your RNAs from your polysomes, and you can then check which ones are being very efficiently translated and which ones are not. This gives you a completely different picture compared to when you would just do microarrays, for instance, on RNA level, right? Because here, your RNAs are all being made. So even if you start with the same amount of RNAs for a certain gene, that does not imply that they are as efficiently translated. And this polysome analysis will give you that added information. That is shown here, and this is my last data slide, and then I'm perfectly on time. What is shown here is essentially cells which are being treated with a drug, and these cells have high RAS or high AKT activity. So that means, so both these oncogenes are high. So these drugs that are being given shut down either RAS signaling or AKT signaling. And then they look at, okay, how many of these, if we do that, and we run these, so the controls, non being treated and the ones being treated, and we run these in microarray, let's say, and we just use the total RNA. We only use total RNA. Then you see that here, that these, in the upper curve, that both these are pretty much like straight lines. And these are, I think, 2-fold or 3-fold differences. There's almost nothing that falls out of these lines. That means, overall, gene expression-wise, shutting down RAS or AKT signaling in these cases doesn't seem to influence that many genes. However, if you do now the same with the polysomal, only with the polysomal fraction, so you do your sucrose gradient and you only use the polysomes and not the ones that are much less efficiently translated, then you see here, both in the RAS and the AKT downregulated profile, that there is here many more dots appearing, and that means that you lose much more genes when you just check the polysomal RNAs. So that means that in this case, these drugs, or the other way around, that if you don't use these drugs, that these influence the formation of polysomes, not the transcription as such, because that is apparently the same, but the way these genes are being translated. So the efficiency of translation is very important. And so both RAS and AKT have a very important function here in recruiting and thus making more or less protein from these polysomes. So that is very important, and that is information that you would never ever get from simple microarrays if you would just do the comparison of your RNA. So that is just an example to show you how relevant these polysomes and how relevant translation can be for a certain phenotype or for outcome or for readout of signaling pathways in a cell. So this is just a summary, and as usual, there should be nothing under that I did not mention. So I think, yes, that's right, 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 and right. And this is also right, I'm pretty sure of that. So this is, I think, just the very last slide, but that doesn't matter because there's nothing new. I think last week you did kind of this, right? All the chromatin and all this epigenetics and transcription. Today you did sort of this. And I think this afternoon Casper will tell you then about when these proteins are there, what happens or what can happen in a cell. So that being said, that's it for now. If you